Good morning to everyone. Today I am going to discuss the basic blotting techniques which are involved in the molecular biology. There are three basic blotting techniques which are oftenly used during experiments that are involved in the molecular biology. The three techniques are southern blotting, northern blotting, and the third one is western blotting. These are called as the basic blotting techniques that are quite commonly used in the molecular biology. The first one, the southern blotting, is used to identify the DNA fragments of interest. Whereas the second one, the northern blotting, it is used to identify the RNA molecules of interest. And the third one is western blotting, which is used to identify the protein of interest. First one, while coming to southern blotting, it was given by, or the experiment was given by Southern Edwin. In this southern blotting technique, first of all, the DNA of interest is identified and later its sequence is identified by different process. And the DNA fragment of interest is identified by this technique by the following process. First of all, if you want to identify the sequence of interest, you can collect either a fragment of DNA or you can collect the whole genome and subject it to restriction digestion. So this is the whole genome of an organism or fragment of DNA. Subject this fragment of DNA to restriction digestion. After subjecting the whole genome or fragment of DNA to restriction digestion, you will get the DNA fragments, which are double-stranded DNA fragments. And before electrophoresis, the double-stranded DNA fragments has to be converted into single-stranded DNA fragments. So this solution which contains the double-stranded DNA fragments, is subjected to alkali treatment or temperature treatment. Such that the double-stranded DNA is converted into single-stranded DNA. Now you get the single-stranded DNA fragments. And these single-stranded DNA fragments are subjected to electrophoresis. In electrophoresis, the DNA fragments are sorted based on the size as well as length of the DNA. The shorter fragments will travel faster in the gel, whereas the larger fragments will be moving slowly in the gel. So after the process of electrophoresis, the DNA fragments, now they are present on the gel, that has to be transferred onto nitrocellulose filter paper. So the gel is collected after electrophoresis and it is transferred onto nitrocellulose filter paper. For this process, nowadays electroblotting is used, where the gel is packed between the nitrocellulose filter papers and electricity is applied such that the DNA fragments which are present on the gel are transferred onto the nitrocellulose filter paper. This is called as electroblotting technique. Or previously it is used, the gel is placed over in between the nitrocellulose filter papers and a weight is put and an alkali mobility liquid is used such that the DNA fragments that are present on the gel are transferred onto the nitrocellulose filter paper. After transfer, The nitrocellulose filter paper now contains the DNA fragments having different lengths. From this, we have to identify our DNA fragment of interest where its sequence can be identified later by DNA sequencing or other methods. Now, to identify our fragment of interest, we use probes. These are short DNA fragments which are labeled. The label may be either enzyme labeled or radio labeled. If it is enzyme labeled, where we can use a substrate to identify the colored band on the nitrocellulose paper. Now this nitrocellulose paper is incubated with probes. As I already mentioned, the probes may be radio labeled or enzyme labeled. If it is a radio labeled probe, you have to subject this nitrocellulose paper, which is attached with your radio labeled probe, to a photographic film, where the photographic film is developed and the band of interest can be identified. Where if you are using a probe which is enzyme labeled, we have to use a colored substrate where band is directly viewed on the nitrocellulose paper, where we can cut out that nitrocellulose paper and we can collect the DNA fragments and we can use it for the further analysis of the or for DNA sequencing. So that is southern blotting. So the next one is northern blotting. So in northern blotting, almost the technique is same as southern blotting, but here the RNA fragments are single stranded. There is no need of these alkali or temperature treatment.